Welcome to the Division of Education, Innovation and Energy e-learning program. I am Ms. Jamilla Bacchus and I am your facilitator for CAPE Biology. Today we are looking at Unit 1 and we are looking at Module 3. We're continuing on the topic of sexual reproduction in animals. So we started on the last day sexual reproduction in animals, looking at the structures of the male and female reproductive system. Today we are continuing on looking at the formation of gametes, this process known as gametogenesis. So gametogenesis in humans. Basically what we are looking at is the way in which we can form the ovum or the egg in females and the sperms in males, the sperms or the spermatogoon in males. So what you should know coming into this topic, you should have known, you should have recalled the process of mitosis, meiosis, and histology. Our objectives today will be to outline the steps involved in oogenesis, outline the steps involved in spermatogenesis, compare these two processes to describe the structure of the human gametes, to be able to draw and label both the human ovary and the Graafian follicle, also to be able to draw, label, and annotate the sperm and the ovum. Lastly, we will want to be able to justify the differences based on their roles and apply the knowledge to answer CAPE-type questions. So, at the end of the day, we are looking to be prepared in answering CXC-type questions. So, one question that can possibly come at you would be to compare the structures and functions of the spermatogoon and the ovum. This has come before, and in many instances, persons will not be specific enough. So we need to be able to look at things including the size, the shape, the contents, the motility, the longevity, and the metabolism between both the male gamete, which is the spermatogoon, and the female gamete, which is the ovum. We also need to be able to justify the differences in size and numbers of the product of gametogenesis. So we have been speaking about the word gametogenesis, and it may be new to some of you, but it's simply the formation of the gametes, the male gametes and the female gametes. We would have met gametogenesis even when we did plants. It simply means the way in which the organism is able to produce the gametes or the sex cells. So here, gametogenesis is the production of sperm, which is known as spermatogenesis, and eggs, which is known as oogenesis. This takes place throughout using the process of mitosis, the processes of mitosis and meiosis. So what is gametogenesis? In oogenesis, you have a diploid cell. Now, let me break it down a bit. When a man's sperm meets with a woman's eggs, when two people get together and they carry out coitus and the sperm swims up to meet with the egg, the nucleus of the sperm can enter the egg and merge with the nucleus of the egg, and thereafter you will have a zygote form. That zygote would have had the nucleus of the sperm, which had N chromosomes, and the nucleus of the egg, which has N chromosomes, and so that zygote is now 2N. It is a diploid structure. Thereafter, that zygote would carry out mitosis and form two cells, and that will continue until you form a whole organism. All the cells of that organism would have been simply replicates of that original zygote, and so they all will be 2N. Now, in the case of the female, what happens may differ slightly to the male. Both of them will start off with these cells which are diploid, which would be 2N. But at some point in time, you have to have the reduction of a cell from 2N to N. Because if that never happens, if that does not happen, then when a woman produces her egg, and that egg is 2N, and if a man produces a sperm and that sperm is 2N, then when they merge together, you will now have an organism that will be 4N. In the case of human beings, that will be what? 46 plus 46 is 92. That is not human. To be human means you have 46 chromosomes in a diploid cell. So at some point in time along the life of humans, you have to have this reduction division called meiosis to reduce the amount of chromosomes. So gametogenesis is the way in which the body is able to break down and produce these gametes, which are half what you will normally get in a diploid cell. So in the female, what you have would be the initial cells in the ovary, which will be diploid oogonium. They go through mitosis until they develop into what we call the primary oocyte, which will then begin the first meiotic division, and then they would be arrested. It will halt for a period of time. It will finish this division and they develop into the follicle, giving rise to a haploid secondary oocyte and a smaller polar body. The secondary oocyte begins the second meiotic division and then arrested again. It will not finish this division unless it is fertilized by a sperm. If this occurs, a mature ovum and another polar body is produced. So this, in a nutshell, speaks to what oogenesis is, and we'll go through it in a stepwise fashion in a little while. In spermatogenesis, on the other hand, there's a slight difference in the way this division takes place. Diploid spermatogonia go through mitosis until they begin development towards becoming gametes. The spermatogonia increases in size and complexity and develops into a primary spermatocyte that will go through the first meiotic division to form two haploid secondary spermatocytes. The secondary spermatocyte will go through a second meiotic division to each produce two spermatids. These cells would eventually develop flagella and become mature sperms or spermatogones. So let's see if we can break it down here. So here we have these cells, right, which would be known as oogonium, right? Plural would be oogonia. These would have been cells in the ovaries. Now at this stage, this is occurring before birth. Before a female child, baby is born, this cell division will be taking place. So you will be having mitosis occurring and you will produce a number of these cells. These will be on the outer layer of the ovary and you will be producing these cells which are diploid. Now, just before birth occurs, some of these cells are going to begin the process of meiosis. And if you recall, meiosis is a type of reduction division that has two stages. It has stage 1, when the homologous chromosomes are separated, and you have stage 2, in which the chromatids of each chromosome would be separated. So just before birth in this female child in the ovary, you will have the beginning of the first stage of meiosis, when the cell would begin prophase 1. The primary oocyte, which is 2N, and in human beings this has 46 chromosomes, this is the cell that the young female baby will have within her ovaries. Okay, this cell it would have started its process of meiosis when that child was still inside of the uterus. And it was halted, meiosis was halted at prophase 1, the first stage of meiosis in prophase 1. However, this will continue, the process of development will continue, but much later. When that child goes into puberty, whether it's from age 9 or age 12, then is when the development will continue in this process of meiosis. So you had a very long halt. Think about it. What could have gone wrong during this process of that, that stalled activity? At puberty, with the release of the hormone FSH, you would have had the continuation of this process. And so this primary oocyte, which is found inside of a structure called a primary follicle, will then begin to develop into what we call the secondary follicle. Within this secondary follicle, you have the secondary oocyte. Now, the secondary oocyte is really at the end of the first stage of meiosis. So you were halted at prophase 1 at birth. And when you reach puberty, you went from prophase 1 to metaphase 1 to anaphase 1 to telophase 1. So cytokinesis 1. And so you would have completed the separation of the homologous chromosomes. What are you going to get? You're going to get 2 cells. But look at these cells. One cell is known as the secondary oocyte. It has half the amount of chromosomes, it is N. And the other cell wouldn't be known as the first polar body. It has the other half of the homologous chromosomes. However, in terms of the size, the first polar body is much smaller because this cell, which really is the one that is going to go into the next, the next generation, this is the potential next individual, because this is the one that has to merge with the sperm. The first polar body is not going to go any further. That is going to decline. That is why most of its cytoplasm actually goes into the secondary oocyte. Now, this secondary oocyte, it reaches to the end of meiosis 1, and then it starts meiosis 2 and is halted at metaphase 2. So when the egg is released, when this structure is released in ovulation, what is actually being released is the secondary oocyte being halted in the stage of metaphase 2, the second stage of meiosis. So it's not yet completed its division, and it's really not yet an ovum. It is a secondary oocyte. When that is released, it may only complete this process if a sperm enters. So if the young lady had sexual intercourse and a sperm would have come up, and the sperm was able to penetrate the several layers and to get to the cytoplasm, it will release its nucleus. And at that point is when the last division of the nucleus will take place. And so you will now go from metaphase 2 onwards to anaphase 2, telophase 2, cytokinesis 2, and therefore you will now have the ovum and the second polar body. So in the ovum, you would expect to see the mature ovum, and in the sides of it, you will see the 2 polar bodies, the first polar body and the second polar body. You will only see 2 because this one never came into being because this did not take any further development. But in truth and in fact, we would think that it would be 4. But in truth but you would have had 3 cells being produced, right? So finally, this cell, which is the ovum, if it meets with an egg, would be produce a zygote, which now goes back to 2n, and therefore we maintain the diploid number. Right? So this whole process of oogenesis was to form the gamete, the female gamete, so that eventually can give rise to a zygote, and so maintain the diploid number of 2n. This simply shows how you have the development from the primordial follicle to the primary follicle, which has the primary follicle, which has the primary oocyte inside of there, then the secondary follicle, which has the secondary oocyte, and the tertiary follicle, which becomes known as the Graafian follicle. What is happening inside of these cells is that you have several cells surrounding this one main cell. So the one main cell, because of its importance in carrying the next generation, it has to be well protected. And so you have several structures surrounding it, protecting it, ensuring that it has enough nutrients, it is well buffered, and so on. And so this structure called the Graafian follicle is what develops. Here we see the structure of the human ovary, and you're supposed to be able to recognize the different structures simply by understanding the anatomy of this ovary. You will be able to recognize the different development that is taking place. So you would see here the primordial follicles. These, as I said, would have been there from before birth. And you would also have the primary follicles. These would be the ones that have been arrested in prophase 1. You then have, so at puberty, these are the ones that will continue developing, and they would be known as the secondary follicle at the end of the first stage of meiosis, and even continuing on to the second stage of meiosis, they will be known as the Graafian follicle. So the Graafian follicle with the egg, the secondary oocyte inside of there that has been arrested in metaphase 2. At the stage of ovulation, you have the secondary oocyte being released, and then is when it can make its way down through the fallopian tube and may be intercepted by a sperm. What happens next is that the remains from this structure known as the Graafian follicle now becomes a new structure called the corpus luteum, and this structure now takes on a different role of actually producing progesterone, and very, very important in maintaining pregnancy, right? So you have the corpus luteum here. It's otherwise known as the yellow body. Obvious because of the color you're seeing. It's known as the yellow body or the corpus luteum, and eventually it breaks down to become the corpus albicans. Good? So you must be able to draw this structure and to be able to recognize and identify the many different stages of development that is taking place in here. Again, this is another drawing of a actual ovary, a cross-section of an ovary. You must be able to recognize the different structures that are present. I know it's a little challenging to recognize, but if you have the skill of observation, you understand the features of the different tissues, you can recognize the structures within here. So you're seeing the secondary follicles here, and you're seeing the primordial follicles at the end. They tend to be more at the end, and you will have the primary follicles. Right? So you're seeing the primordial follicles are very, very small. Remember, these are the ones that would have been before you actually had the first stage of meiosis beginning. The primary follicles are the ones where you had the cells being arrested in prophase 1. The secondary follicles would be the ones continuing on to become the secondary oocyte. Here you see the mature Graafian follicle. You are supposed to be able to draw and identify the structures within this Graafian follicle. This here is the structure that is carrying the next egg that could potentially be the next organism. And here in the middle of it is where you have the secondary oocyte. The secondary oocyte is well protected, as you see, by this layer here called the corona radiata. And there's a layer that is around it called the zona pellucida, a clear layer around it that seeks to buffer but also helps with communication between it. Around, you have the antrum, the follicular fluid, and this fluid does a very important role of buffering, protecting, and also having some enzymatic activity there. You also have this layer surrounded. These cells are what you call granulosa cells. They play a very important role in producing estrogen, which we know is produced in the ovaries and play a very important role in development of the ovary. In development of the uterus. The uterus, the estrogen helps the uterus to produce the lining that will, if implantation does take place, it will have a nice place to be buffered to prevent from external shocks and so on. So this Graafian follicle, know the difference in the structure, the different parts of the Graafian follicle. This outer layer is the layer called the theca, again having granulosa cells in that region, right? So you just need to have an appreciation of this structure. This here is the ovum. As you can see, you're recognizing that you have two polar bodies there. How do we know the difference between a secondary oocyte and the ovum? The secondary oocyte would have had one polar body in it. The ovum now has two polar bodies because this is the final stage when a sperm would have entered and the last stage of development of the nucleus took place, and so you had the second polar body, right? Here you are seeing the two nuclei from the, you're seeing the two nuclei, one from the male and one from the female. They are called the pronuclei, and so at this stage now, you would have had a zygote forming because the both nuclei would have merged, right? So this here is the first cell that would be produced, called the zygote. Thereafter, this cell would carry out mitosis, and you will have the beginnings of an embryo. Embryo is a tissue of a mass of undifferentiated cells forming a tissue, and thereafter that embryo will develop into a fetus and then a baby. So this is showing again what is happening within an egg. This one would have been before the cell division. So again, you can differentiate whether it is whether it is a secondary oocyte or an ovum simply by looking at whether you have one polar body or you have two, as we saw a little while ago. So this one, it has not reached in yet to release its nucleus, so this nucleus has not yet completed the division, so you only have one polar body. Good. So what I want you to do would be to look at these structures and be able to go and research what is the function of the zona pellucida. What is the function? The zona pellucida is that region, that jelly-like region just surrounding the cell membrane of the secondary oocyte. And what is the function of it? What is the function of the corona radiata? The granulosa cells, the theca, and the antrum. I would like you to go and research and be able to, show the relationship between the structure of these tissues and their functions. In the males, spermatogenesis is occurring. Now, as I said, there are some similarities and some differences between both spermatogenesis and oogenesis. Here we are seeing that this process also involves mitosis and meiosis, similar to what happened with the female. Remember, in the female, we started off with the oogonia carrying out mitosis, and so increasing the number of cells. Here too, you have the spermatogonia. Singular is spermatogonium, plural is spermatogonia. the spermatogonia would have also carried out mitosis to continue to produce more. If a man stops carrying out mitosis, then he will no longer have any sperms. So he's constantly carrying out mitosis, whereas the woman ceases. Once a woman is born, you would have had mitosis ceasing with her oogonia, while the man, he continues to carry out cell division mitosis. So you would have had the spermatogonia in the outer region. So in this here is showing one of these structures. This structure is known as the seminiferous tubule, and the outer area is where you have these cells that would be what we call the primordial cells. These are the cells that will be carrying out mitosis to continue to have a supply, continue to have an increase. Just as how your skin is constantly producing new cells, so too at the outer area, you're constantly producing new spermatogonia. Now, these will carry out cell division, increase their numbers, and at some point, they are going to begin to develop. Some of them will develop, while some will still be carrying out the cell division. They will develop and reach the point called the primary spermatocyte. At this stage, they are ready to begin the process of meiosis. Remember, there are two processes. There is mitosis and there is meiosis. Don't get confused with the pronunciation or the spelling. Whether you say meiosis or meiosis, some people say it. Understand, meiosis is the one where you have the reduction division. Mitosis is the one where you have the replication division. So why is meiosis important? Remember, for us to be able to to go from 2n, 2n stands for 2 times the number of different chromosomes. Remember, we would have done that in mitosis since in unit 1. And to go to the point where we have n chromosomes, to go from here to here, we had to have a reduction division called meiosis. So this is what is happening here inside of the testes, as we saw happen with the eggs. So we start off with the primary spermatocytes. These would have carried out the first stage of meiosis and ended up with the secondary spermatocytes. And so these cells are already N, because at the end of the first stage of meiosis, we separate the homologous chromosomes, and so we have already reached to N chromosomes, 1 time the number of different chromosomes. Thereafter, this simply continues with the second stage of meiosis, and so these are also N, but these are called spermatids. They are not yet fully sperms. They are spermatids, and the next stage is they will get their tails, and they will now be known as the mature sperm or the spermatogoon. But between these different divisions, you have a very important process that is happening. They have to identify and interact with a cell called a Sotoli cell. The Sotoli cell plays a very important role in ensuring this process continues and ensuring these sperms are nourished. So the Sotoli cells, these are found in areas throughout, okay? So you're seeing the Sotoli cells here. These cells are found in regions interspersed. They're interspersed between these cells, and they provide nourishment for these sperm cells to allow them to reach to full maturity. When we come in the next lesson to the way in which hormones control this process, you will see what is the role of testosterone. Testosterone plays a very important role in ensuring the Sotoli cells work effectively. So someone who has a problem with production of testosterone may have a problem with sperm production. And that's why we keep saying one thing will also facilitate or impede another thing. So if a man has a problem with production of testosterone, he will have a problem with sperm production. So these Sotoli cells, we should see them in another way. So this cell here is the Sotoli cell. As we can see, this is the nucleus of the Sotoli cell here, right? And we can see these different cells. the primary spermatocyte. This would be the primary spermatocyte. And it is interfacing with the Sotoli cell. So this here, this whole thing here is the Sotoli cell. But these other stages of the development, they have to interface because they're getting nourishment from the cell, right? So these cells are developing, and the final development takes place where the tail is more or less to the middle, and so you will see the tails tend to be out towards the end here, more or less to the middle of the seminiferous tubule. Okay? So if you see here, see how the tails are out. So this is the middle area of the lumen, and the tails are hanging out there. So the development takes place from here coming in. So the sperm cells will be inside of here. The final product will be in the middle, and so they just go along, and the middle will be hollow, and they just go along in the middle, and they swim along, and they will go to the epididymis where they are stored. So they are produced inside of the structure called the seminiferous tubules. And this is showing a cross-section of a seminiferous tubule. You are supposed to be able to draw one of these. For CXC, purpose of CXC, you're supposed to be able to explain what is happening here, and you're supposed to be able to draw a cross-section of these structures. Right? So here, I would like you to sit and to draw a cross-section. And to make it easy for you, as your teacher may do in school, I would tell you to do a pie segment. So don't do the whole thing, because I don't want you to just draw circles, circles, circles. I want you to do an honest, realistic drawing of what you will see. So you can go online, look at a picture of a cross-section of a seminiferous tubule, and simply draw. You use your ruler, and you pull a straight line like this. That is a pie segment, and you're drawing what you see inside of here. So if you're seeing cells and you're out here, you draw it. If you see a Sotoli cell in this area here, practice drawing it, please, because you can get that for exams, and then you will see the cells, right? So I'm just giving you that. Obviously my drawing is not ideal, but I'm just telling you, practice doing these drawings, please. You can just do a pie segment, because we prefer that you do a small section and you make sure, like you don't do like what I do there, you make sure your circles are perfectly round, that I can't see any space or anything that is showing. Lastly, we just like to look at the human sperm. You're supposed to be able to know the structure of the sperm, just as how we're supposed to know the structure of the Graafian follicle or the secondary oocyte. This is the male gamete, and we are supposed to also know the structure of the sperm. Note, the sperm has its main area here, where you have the main part of the cell. This is where you have the cytoplasm, you have the nucleus, and you have a whole lot of different organelles in here. To the front part, you have the acrosome. This area here is known as the head. This is the neck. So this area here is the neck, right? This area here is the midpiece, and this area here is the tail. The tail has an important role to play in swimming, and so it has a lot of microtubules. It's in an arrangement of 9 to 2. So if we check it, we will see 9, 1, 2, 3, 4, 5, 6, 7, 8, 9, and you have 2 in the middle. So you have a 9 to 2 arrangement of the microtubules. They play a very important role, providing mechanical strength to allow the sperm to be able to swim. If we look at our hand, and I do like that, you would appreciate that for my wrist to move, for my hand to move, is really my wrist that is moving. So for the sperm to move, this part here, where you have a lot of mitochondria, a lot of energy here, is allowing it to be able to swim. Okay, so we have done a lot today, but I'm sure you can appreciate the structures of the spermatogoon and the ovum. In terms of the size, the egg is approximately, it's the largest cell in the human body, the egg cell. It could be as large as 0.3 millimeters, and while the sperm could be about 0.3 micrometers. So in terms of the size, the sperm is much smaller than the egg. In terms of its shape, the egg is round. It actually, it's perfectly round, while the sperm is elongated. And of course, when you look at the head of the sperm, that torpedo-like shape is to allow it to be able to maneuver and to move with least amount of resistance through the passageways. And when we look at the contents within the sperm and within the egg, we would appreciate the egg contains much more than the sperm width, but however, the sperm has a lot of mitochondria for its the ability to swim through the mucus and so on. The motility, the sperm is much more active in terms of swimming and so on. The egg actually cannot move on its own. That is why the fallopian tube has cilia to be able to sweep it along. The longevity, the egg lasts much longer than the sperm, even though the sperm is going to remain viable for longer. The egg is only viable for 24 hours, while the sperm is viable for 48 hours. But in terms of the longevity of the egg is what will remain and be able to go on to form the new individual, while the sperm will die. Metabolism, the egg is more metabolically active, while the sperm actually, it is not as active. Justify the difference in size and the number of products of gametogenesis. Obviously, the egg, you don't want a lot of egg. You want to concentrate your effort in producing one good egg. So during the process of meiosis, you only have one egg developing out of a potential 4 at the end of meiosis. And so the organism is able to concentrate its efforts, its energy on producing a good gamete. While the sperm, because of the chances of reaching the egg is so there are so many things working against it, not to mention things like the pH, the amount of crevices that are between the different structures, the cilia, the distance you have to travel. Obviously, because you have all these things working against it, you have to have a lot more sperm, the size and all of that, the storage. You have a lot more sperm than you will have egg, right? So you need to be able, you must be able to justify why you have the differences in the sizes and the numbers of the both gametes in gametogenesis. Good. So we would have gone through a number of the objectives. We would have met the objectives that we set out to outline the steps involved in oogenesis and spermatogenesis, to be able to compare the processes. I would like you to really sit and make a chart to compare both processes in terms of the length of time it takes, in terms of the divisions that are happening, whether there is a halt in the division or not. I would like you to sit and compare what is happening in both processes, because you may be asked to do that. Describe the structure of the human gametes, draw and label the gametes and all the structures, and also justify the differences and answer questions. So I know this topic, again, it may seem overwhelming for some persons. I know some students, they think that the process of the cell division is a bit confusing. What you need to do is to slow down, take it one step at a time. I think many of you have problems because you would have forgotten the processes of mitosis and meiosis. So I would suggest to you that you go back there, break it down, go back over, especially meiosis, what is happening, what are the different stages of meiosis, and you should be able to do better in explaining the process of oogenesis and spermatogenesis. I am certain that you will be doing very well as you go along, and I look forward to continuing this process with you. On the next occasion, we'll be looking at the way in which hormones control the process of gametogenesis. Have a pleasant day. Bye.